Hello, everyone. My name is Austin Adkins. I am a first year PhD student working in the sleep lab under Dr. Sanford and Dr. Wellman. Today I'm going to be telling you about the work in my master's thesis while in this lab, something I think we all can relate to: stress, sleep loss, and how fear memory affects neuroinflammation as it pertains to these mechanisms. Stress can alter brain function, disrupting subsequent sleep and triggering neuroinflammation. These symptoms are implicated in a number of fear and anxiety based psychiatric disorders. Unfortunately, the persistence and intrusion of fear memories related to stressful experiences is quite common and can inappropriately produce these symptoms. If neuroinflammation persists, normal brain function can become altered. Previous studies in our lab have demonstrated differential effects of stressor controllability based on controllable or escapable stress and uncontrollable or inescapable stress on sleep following stress and fear memory recall. However, it is unknown how fear memories influence neuroinflammatory processes and the relationship between neuroinflammation and sleep. There is a need for better understanding these mechanisms and how they are connected, and how fear memories influence them following a stressful or traumatic event. In this study, we determine how fear memories associated with escapable stress and inescapable stress impact the neuroinflammatory response and its relationship to sleep. First, we implanted mice with wireless telemetry transmitters for recording of sleep and whole body temperature and gross body movement. Following a recovery period, a 20-hour baseline sleep recording was obtained. Then, mice were divided into groups. ES and IS groups were exposed to the shuttle box where they underwent shock training. Animals exposed to ES had the ability to learn if they moved across the chamber to the other side, they could behaviorally terminate the shock. Animals exposed to IS had no opportunity to terminate the shock. A home cage group, where the mice were never removed from their cage for the entire study, was used as a control. Following training, another sleep recording of 20 hours was obtained in all groups. 1 week following shock training, to assess the effects of fear memory, ES and IS groups were re-exposed to the shuttle box but did not receive foot shock. This evokes contextual fear in shock-trained animals. Following context, sleep was recorded in all groups for 2 hours. All shock training and context sessions were video recorded for scoring of freezing, a measure of behavioral fear in rodents. After the recording, mice were perfused with PBS, their brains removed, and regions of interest, the hippocampus and amygdala, microdissected. RNA was isolated from each region, and mRNA expression levels of inflammatory-related genes were measured using nanostring panels. Baseline REM amounts did not significantly differ between ES and IS groups for any parameter examined. Analysis of total REM revealed ES mice exhibited overall significantly greater REM across the recording periods, while IS mice exhibited less REM. Baseline non-REM amounts did not significantly differ between ES and IS groups for any non-REM parameter examined. The only significant differences occurred over the 20-hour recording during the dark period, where ES mice exhibited significantly greater non-REM following shock training 1 and 2. This contributed to the overall greater non-REM amount within the ES mice compared to the IS mice over the total 20-hour recording period. Analysis of gene expression revealed distinctly different expression levels between ES and IS groups when compared to the home cage control. Within the hippocampus, ES mice showed a downregulation of many genes associated with neuronal damage, immune cell recruitment and activation, and inflammation. They also showed an upregulation of genes associated with the protection of neuronal cells. Compared to home cage, IS showed a downregulation of genes associated with neuronal protection. They also showed an upregulation of genes involved in cytokine signaling, immune cell activation, and pro-inflammatory pathways of the innate immune response. Within the amygdala, ES mice showed an upregulation of genes involved in neuronal signaling and the suppression of protein aggregation and nerve damage. IS mice showed a downregulation of genes associated with neuronal signaling and the myelination of neurons, while there was an upregulation of genes involved in pro-inflammatory T-cell signaling and protein aggregation within the IS group. Here, we have demonstrated that the sleep and immune signaling pathways communicate and are influenced by fear and stress systems, though the precise mechanisms that mediate these influences are unclear. Stressor controllability differentially influences the subsequent responses of the sleep and neuroimmune mechanisms. Persistent fear memories following stress exposure may facilitate immune activity in the CNS, influencing neurotransmitter systems, neurocircuitry, and behavior, as seen in the gene array results. While out of the scope of this study, cytokines may be largely contributing to the maintenance of fear and anxiety-based symptoms by affecting the activity and connections of brain regions implicated and the etiology of these disorders, including the amygdala and hippocampus. Proper emotional processing and extinction learning may be responsible for the suppression of inflammatory processes and prevention of sleep loss, which may serve as a protective coping mechanism. Further studies should be conducted that delineate the mechanisms that regulate the relationship between sleep, neuroinflammation, and fear behavior. Therefore, future studies should focus on integrating systems when studying sleep and neuroinflammatory responses to stress in order to fully reveal how these mechanisms function together.